Cultural Academy welcomes you all to online classes for horticulture. Friends, if you are interested in online tests and to get the PDF materials, you may take down this mobile number to call and do WhatsApp, and you may do mail to this mail ID too. Friends, kindly subscribe the channel to receive the upcoming videos. Friends, today we are going to see chapter 15 from Unit 3, Micropropagation and Micrografting. First, we are going to see about micropropagation, which is a part of biotechnology. Controlled use of biological agents such as microorganisms or cellular components for beneficial use. Development and utilization of biological processes, forms and systems for maximum benefits to man and other forms of life. Now, let us see some important biotechnological institutes in India. International Center for Genetic Engineering and Biotechnology is located in India and as well as in Italy. In addition, the research centers for Lal Bahadur Shastri Center for Advanced Research in Biotechnology is located at Indian Agricultural Research Institute, New Delhi. National Dairy Research Institute, Karnal. Indian Veterinary Research Institute is at Naham. The Biotechnology Center of Indian Agricultural Research Institute is called as National Research Center for Plant Biotechnology. Plant biotechnology aims at improving the genetic makeup, phenotypic performance or multiplication rates of economic plants. There are two basic techniques used in plant biotechnology. They are plant tissue culture and genetic engineering. First, we are going to see about plant tissue culture. It is the cultivation of plant organs, tissues or cells in vessels using artificial media, and the technique of plant cells and tissue culture are also called as in vitro technique. Friends, in history of plant tissue culture, scientists and their contribution is most important one. So, please have a look on it. Let us see some terminologies in plant tissue culture. Totipotency. The regeneration capacity or ability of a plant cell to develop into a whole plant is known as totipotency. Callus. A mass of unorganized regenerated cells in culture medium is called as callus. De-differentiation. The conversion of mature cells into meristematic state leading to the formation of callus. Redifferentiation. The process of biochemical and structural changes by which cells, especially the unorganized cells, becoming specialized in the form and function with the ability to form a whole plant. Protoplast, cell without cell wall. Now we are going to see what are the techniques to be followed to raise a tissue culture plant. First, we have to select an explant for in vitro culture. Explant may be a tissue or organ excised from a healthy plant. And then we have to do surface sterilization to eliminate bacteria and fungi with 1 to 2% solution of sodium or calcium hypochlorite or 0.1% solution of mercury chloride. Rinse the explant in sterilized distilled water to remove the disinfectant. We have to do all these above procedures under laminar airflow chamber to get rid of the contamination. We have to do sterilization to inactivate the microbes present in the culture media, culture vessels and instruments. After sterilization, plant cells and organs are cultured in nutrient medium. This nutrient medium contains inorganic salts, vitamins and sucrose as a carbon source, and it also needs some growth regulators for root formation, auxin, and cytokinin for shoot formation. Auxins are 2,4-D, NAA and IAA, kinetin and benzyl aminopurine. Commonly used is kinetin zeatin. Organic supplements like coconut water, seasoned hydrolysate and yeast extract. pH of the medium should be 5.5. Solidifying agent is agar. Commonly used media is MS media. Callus culture. We have already seen, but once again I am reminding you, the cells on the agar medium develop into an unorganized mass is known as callus. Then what is about suspension culture? In liquid medium, a suspension of free cells and small cell masses. The medium is autoclaved at 15 psi for 15 to 20 minutes. Environmental conditions. The organ and cell culture are evaluated under controlled temperature, light and relative humidity. Temperature should be 18 to 25 degrees Celsius. Light is not essential but beneficial for plantlet regeneration. And for relative humidity, the plantlet should be kept in culture room or in incubator. Subculturing. After a period of time, it is necessary to transfer organs and tissue to fresh media. In tissue and cell culture, culture portion of tissue is used to inoculated in new culture tubes. Subculture of Callus culture every 4 to 6 weeks and suspension culture every 3 to 4 days. Friends, now we are going to see plant regeneration and transfer to soil. Production of shoots and roots from cell and tissue culture is known as organ regeneration or organogenesis. Transfer of whole plants from test tube to soil is easy, but before transferring, the plants should be pre-treated to make them hardy. In laboratory, they have transferred in small pots and covered with vessels, inverted beakers to prevent excess transpiration. After 3 to 4 days, covers are removed and kept in diffuse light for 5 to 10 days. Hardening on large scale is done by using mist chamber. Plants then transferred into greenhouse and after 1 to 2 weeks planted in soil and kept in sunlight. Let's see the stages of micropropagation. There are 4 stages of micropropagation. First, we are going to see about stage 0. This is the initial step of micropropagation in which stock plants have to be grown under controlled condition before using for culture initiation. Stage 1. Preparation of explants from stock plants is followed by its establishment in a suitable culture medium. The steps involved in stage 1 are explant isolation, surface sterilization, washing, and establishment of explant on appropriate culture medium. Stage 2. This stage involves the multiplication of shoots or rapid somatic embryo formation using a defined culture medium. The harvest cycle generally takes 4 to 8 weeks. Either the shoots are marketed directly or carried out over the next stage for further development. Stage 3. Shoots obtained from stage 2 are transferred to the next rooting or storage medium. These shoots are directly established in soil as a micro cuttings to develop roots. The shoot handling at this stage differs from species to species. Transfer of plantlets to sterilized soil for hardening under greenhouse environment is achieved. This step is to ensure the successful transfer of plantlets of stage 3 or unrooted shoot apexes of stage 2 into a suitable compound mixture or soil in parts under controlled conditions of light, temperature, and humidity. For marketing, sometimes these plantlets are established in an artificial growing medium such as soilless mixtures, rockwood plugs, or the sponges. It takes 4 to 6 weeks for marketing of the finished products. Friends, we have completed stages of micropropagation and now we are going to see classification of plant tissue culture techniques. Based on the plant pots used as explants and type of development in vitro, it is classified into 4 types: embryo culture, meristem culture, anther or pollen culture, tissue and cell culture. Embryo culture. Young embryos are excised from developing seeds and are placed on a suitable nutrient medium to obtain seedlings. 2 types of embryo cultures are used. Culture of immature embryos, that is hybrid seed, example soybean, barley. Culture of mature embryos, example orchids, iris, etc. Now we are going to see meristem culture, which is widely used for quick vegetative propagation of large number of plants. We have to take 5 to 10 mm shoot apical with leaf primordia and regeneration takes place in a suitable culture medium. It involves pre-existing apical meristem and regeneration of advantageous roots. Anther culture is otherwise known as pollen or microspore culture. Haploid plants may be obtained from pollen grains by placing anthers or isolated pollen grains on a suitable culture medium. For successful anther culture, plants should be grown under controlled temperature, light, and humidity. Tissue and cell cultures. Application of plant tissue and cell culture in crop improvement depends on the technique for regeneration of whole plants from them. Both callus and suspension cultures of many species regenerate whole plants. Somatic embryos regenerate to form whole plants. Example for this is carrot. In shoot regeneration, shoot buds differentiate from the cell mass followed by differentiation of roots. Example for this is coffee. Let's see about shoot regeneration. Shoot buds differentiate from cell culture. Shoot buds are unipolar, that is, have only primule and have vascular connections with the callus tissue. High cytokinin to auxin ratio supports shoot regeneration, while high auxin to cytokinin ratio promotes root regeneration. Friends, keep it in your mind. Gibberellins suppress shoot regeneration. Somatic embryogenesis. Somatic embryos originate from single peripheral or deep-seated cells of Callus. They form group of cells and it divides and progresses through globular, heart-shaped, torpedo-shaped and cotyledonary stages similar to zygotic embryos. Now we are going to see somatic hybridization. Somatic cells are manipulated by removing the cell wall and protoplast are fused. This process of hybridization at cell level is considered as an alternative to sex. Usually, sexual hybridization has been used for transferring of various agricultural important traits into numerous cultivated crops. An alternative system for hybridization of plants is somatic cell hybridization, which is otherwise known as parasexual hybridization. Hybrids, it is otherwise known as cytoplasmic hybrids or cells containing nucleus of one species but cytoplasm from both the parental species. Let's see the one-liner for this topic. Which scientist had their contribution in 1962 for the development of haploids through anther and pollen culture for the first time? Guha and Maheshwari. Suspension culture. In liquid medium, a suspension of free cells and small cell masses. The medium is autoclaved at 15 psi for 15 to 20 minutes. Shoot buds are unipolar, that is, have only primule and have vascular connections with the callus tissue, is known as shoot regeneration. Let us see what are the growth regulators to be used in nutrient medium. For root formation, auxin and cytokinin for shoot formation. Somatic cells are manipulated by removing the cell wall and protoplast are fused. It is known as somatic hybridization. Hybrids, which is otherwise known as cytoplasmic hybrids, are cells containing nucleus of one species but cytoplasm from both the parental species. Friends, we have completed chapter 15 from unit 3 and now we are going to see the important questions and answers from this topic. What are the macronutrients used in plant cell culture medium? Nitrogen, phosphorus, potassium, sulfur and calcium. Which is the most common carbon source used in the plant cell culture medium? Sucrose. Silver thiosulfate is added to culture medium as it helps to prevent the gaseous plant hormone ethylene dioxide from accumulating to detrimental condition. Which one of the following is not a cryoprotectant? Potassium chloride. Friends, you may contact through these two communication ways to get the PDF material and online testers. Thank you for watching the video. Kindly subscribe the channel to receive the upcoming videos. Thank you.